Hello everyone, welcome back to my channel. This is my third video. If you have been following me or if you are a subscriber of my channel, you must have seen my second video. And if you have not seen my second video, I request or I urge you to go and check out my second video. The topic was about the staining of the bacteria. So related to that topic, this is the continuation of the second video, and the topic for today learning is microscopic methods used in the study of microorganisms. As I have already explained in the second video, that the purpose of staining is done to enhance the visualization, or we can say to clearly visualize the microorganisms while observing it under the microscope. So stains, S-T-A-I-N, stains basically means in layman terms we can say we color it or we dye the microorganisms after fixation, so to enhance the visualization of those microorganisms. So basically there are two types of staining. First is your vital staining and second one is the supravital staining. In vital staining, what we does is the microorganisms are not killed, but in supravital organisms what is does is the microorganisms here are killed. The microorganisms could be virus, bacteria, fungi, parasites. So what we does is we kill the microorganisms and after fixation we stain it with the dyes. So as you know, in supravital staining there are four methods or techniques. The first one is the simple staining, the second one is the negative staining, the third one is the impregnation method, and your fourth is the differential staining. So under differential staining comes the gram staining, acid fast staining, or also known as Ziehl-Neelsen method, and third is your Albert staining. So related to those topics, there are mainly two types of preparations. So one is the unstained preparation and second one is the stained preparation. So what we does is for the study of these microorganisms, we stain the bacteria, or sometimes what we do is we do not stain the bacteria. So the first topic is the unstained preparation. As the name itself implies, here what happens is there is no usage of stains. Stains, as we know can be acidic in nature, basic in nature, or the neutral in nature. So under unstained preparation, you came to know about that there is no usage of stains. So under this unstained preparation, there is first technique is the wet mount method and second one is the hanging drop method. So in wet mount method, what we do is we take a glass slide. And with the help of an inoculating loop, we take the sample or the pathological specimens. So this is your sample or your specimens. So these samples could be your urine, stool, pus, or sputum. So it could be any kind of discharges. So after taking the specimen with the help of an inoculating loop, we put it on a glass slide. We have put the sample on the glass slide. So the second step is what we does is we take a cover slip. As you know that glass slide is rectangular in shape. The easiest way to distinguish both the slide is by your glass slide is rectangular in shape while your cover slip is square in your shape. So after putting your specimens on the glass slide, what we do is we take the cover slip which is square in shape and put it above the glass slide. So if we put the glass slide above the if we put the cover slip above the glass slide, what it will somehow look like this. Okay, so it will somehow look like this. So after putting it the cover slide on the glass slide, cover slip on the glass slide, what we do is we mount it on the microscope and then we observe it under. So first we observed it under low power, and then second under high power, to increase magnification. So you're clear with your wet mount method. The uses of wet mount method is to observe or to examine the presence of the microorganisms, of the particular microorganisms. So this is your wet mount method or your wet mount technique. And second comes your hanging drop method. It is slightly different from the wet mount method. So here what we take is, here we first take a cover slip. Okay, here what we do is we first take a cover slip, put the pathological symptoms same way, your urine, stool, pus or sputum. We take the samples and put it on the cover slip with the help of an inoculating glove. After putting it on the cover slip, what we do is we next take a glass slide and put it over the cover slip. So how it will look like? It will somehow look like this. I'm not doing it practically, so I am drawing it for your better understanding. So after putting it above the glass slide, what we do is we then lift it and turn it upwards, meaning after we lift both the cover slips and the glass slide and keep it backwards. So what happens is your cover slip comes on top of it and your glass slide is on the bottom side. Initially the cover slip was on the bottom side while your glass slide was on the top side, but after lifting and inverting it, the cover slip goes on the top side and your glass slide comes to the bottoms. So what happens is your sample was on the cover slip. So when we flip it downwards, what happens is the cover slip stays at the top of your glass slide and your specimens or your sample hangs above your glass slide. So this is why this method is called the hanging drop method. So after lifting it, what we do is we mount it on a microscope and observe it in the same process. First is under low power and then high power. So the uses of hanging drop method is to study the motility of organisms. So when we focus on the edge of the drop, we can see the motile organisms. So this is your unstained preparation. The first is your wet mount preparation, and your second one is the hanging drop method, and second comes your stained preparation.